Hey there, welcome to the second lecture on the chapter Human Health and Diseases. In this lesson, we'll be basically dealing with the topics like attack on a pathogen, the parts of the immune system, different kinds of immunoglobulins, and where the fighters of the immune system are formed. With fighters, I mean the elements of the immune system. Okay, so let's begin the lecture. Yes, so here we are at the site scenario. Okay, now in bone marrow, what is bone marrow? I think you are already clear about. Bone marrow is the part of the body where RBC formation takes place. Okay, where blood cell production takes place. Now, in bone marrow, there are certain cells called as stem cells. Stem cells are those which have the capability of forming into anything. A stem cell can form your muscles of your leg, it can form your cardiac muscles or anything. That's the beauty of those muscles, alright. Now, there are certain specific cells called as lymphocyte precursors. Okay, what is lympho? Lympho, wherever comes, just understand it is in regard with your immune system. Okay, cyto means cell, precursors means primitive of something, like they are going to form something. That is their primitive version. Okay, so these lymphocyte precursors are inside bone marrow, that are kind of stem cells, are of further two types. T cells, they form these two actually. T cells and B cells. Okay, T cells where T stands for thymus and B cells where B stands for bone marrow. Okay, T cells have further types: memory cells, killer cells, suppressor cells, and helper T cells. Okay, and B cells have types: memory cells, plasma cells. Okay, that plasma cells form antibodies. I'll be discussing that later on. Okay. Here T stands for thymus in T cells and B for bone marrow. Now just understand the difference. The naming is different and the memory cells both have in common. Okay, the naming is different just on basis of where they get matured. Okay, the T cells and B cells, since both are formed in bone marrow, have same origin, but the T cells get matured in the thymus gland. I think you know where the thymus gland is. Yes, exactly. It is located where the windpipe branches. Okay, branches one towards right lung and one towards left lung. At that particular point of division, thymus gland is situated. Okay, so the maturation of the cells from bone marrow takes place in T cells, and the cells under it are said to be T cells. Okay, and in B cells, the both maturation and formation takes place inside bone marrow, so B cells. All right, the cells in T cells form the cell mediated immunity, and the cells in B cells form the antibody mediated immunity. Okay, in the previous lecture I talked about something called as memory of the immune system. For that memory, the cells responsible, here you can see memory cells, that I'll be discussing it later on. Okay, so moving on to the next slide. Yes, what I was speaking to you, like all these T and B cells are formed in bone marrow. Okay, the difference is that the T cells migrate to thymus and their further development takes place there. Okay, is that correct? Clear? Be clear about this because you might get a question regarding this. Next, we are having cell mediated immune system. At first we are having helper T cells, then killer cells, then suppressor cells, and then memory cells. Helper T cells, understand it, they stimulate killer T cells and B cells to divide. Now, helper T cells means obviously they will be helping someone. Someone means here cells. Okay, now they basically stimulate the killer cells to divide. Why to divide? Why to divide? Obviously to get increase in their number. All right, so their main role is for stimulation of those cells. Okay, one more thing, they produce lymphokines. I told you about lympho. Yeah, remember that? Okay, lymphokines are certain chemical substances which are secreted and that are meant to attract phagocytes. What is phagocyte? I think you are clear about it. What is phago? Phago means to eat up, and cyte means, I told you cyto, yeah, it's a part of that. Cytes means cell. So to eat up cells. Okay, so that is a phagocyte. So to attract a phagocyte, they produce lymphokines. I'll be telling why to attract a phagocyte. Okay, next, killer T cells. Killer T cells basically are destroyers. They kill up. Okay. They secrete lymphotoxins that toxicate the pathogen. Here we'll be referring to bacterium, say for our ease, as the pathogen. Okay. They secrete toxic substance to kill that bacterium that has entered. Okay. Then we are having lymphokines, as I mentioned earlier. Now just understand what is the use of this lymphokine. Why? To attract a phagocyte. Suppose a helper T cell is moving, it encounters a pathogen. Now since it is a helper cell, it cannot kill that pathogen. So what it will do is that it will produce some lymphokine which will attract the phagocyte, the cell eater of the immune system. So it will get attracted to that lymphokine, it will come there and eat up that pathogen. So this is the role of what? A lymphokine. Okay, clear about that? Next, the very important suppressor T cells. Now, suppressor T cells function. Okay, suppressor T cells function is to suppress the activity of other cells of the immune system. Understand that. That is to protect action against own body. That is very, very important. Sometimes what happens is that our immune system fails to identify our own cells and it starts attacking our own body cells, considering them to be pathogens. So that is said to be an autoimmune disorder. We will be discussing it further in further slides. Okay. Now, the role of these suppressor T cells is to suppress the activity of the immune system cells to that extent so that we can stay protected in case of an autoimmune disorder. So this is the role of suppressor T cells. Okay. Memory T cells, obviously they store the activity of the immune cells at the time of an encounter so that the next response will be much more stronger. Okay, clear about it? Okay, so moving to next slide. Next we are having antibody mediated immune system. I told you regarding which elements are there. Obviously the B lymphocytes. The plasma cells, as I mentioned in the first slide, form immunoglobulins that become a part of antibody mediated immune system. Regarding the diagram, I am telling you, stick to your NCERT textbook. Just understand the two big chains. As you can see, just wait, I'll show you. Yeah, these two big chains form the heavy chain and these two form the light chain. Okay, and these are connected by disulfide bridges. Just understand, just be clear about how the diagram is labeled. Just be clear about it. Okay, I'll be discussing you what is important in this in further slides. Okay. Immunoglobulin, there are further types of it, I'll be discussing it. The components, I think you are clear. Okay, that is two heavy chain, two light chain and bonded via disulfide bridges. Okay, yeah, one more thing, this antibody mediated immune system is also said to be humoral immunity. That's very important. Why humoral immunity? Because humoral means in regard to fluids of the body, and this is as a part of that called as humoral immunity. Clear about it? Antibody mediated immune system is also known as humoral immunity. Clear? Now moving to types of immunoglobulins. Okay, oh sorry, before that, how a immunoglobulin works. Consider a bacteria as a pathogen. Okay, now what is going to happen is that there will be at first agglutination. In this process agglutination, the antibody, just understand what is an antibody. I think you are clear. Antigen, that is a pathogen. Now what we are going to discuss is antigen antibody interaction. Antigen is the pathogen, antibody is the one that is formed inside your body and they are going to fight. Okay, just enjoy the fight. How it happens, just learn about it. At first there will be agglutination, that is the attachment of antibody on the surface of the pathogen or the antigen. Suppose here the bacterium is the pathogen. Okay, so the antibody goes and attaches to the surface. This process is agglutination. Clear? Next, opsonization, that is now it gets attached, now it will start secreting some substance called as opsonin. Okay, that is basically composed of IgG and IgM. I'll be discussing it in the next slide. Okay, just have patience. What will this do? This will facilitate the phagocytosis process. I told you about phagocytosis. Okay, phagocytes I told you. Phagocytosis is the process of phagocyting something. Okay, then neutralization. In neutralization, the toxin in the bacterium will be neutralized. So this is the process. Remember it: agglutination, opsonization, neutralization. These kind of questions you will be sure shot getting in AIMS, because AIMS focuses on human physiology and health and diseases. Okay. So next slide. Yeah, this is very important. I'm telling you. Immunoglobulin types. Okay. At first, immunoglobulin A. Okay, immunoglobulin A that are 10%. Okay, in amount they are 10%. They protect the mucous membrane and from inhaled pathogens. They are in colostrum, saliva, sweat, tears. This is a very common question. Which immunoglobulin is found in colostrum? I think you know what colostrum is. It is the yellowish milk secreted by a lactating mother after the delivery of the child. I think it's a bit complicated now. I'll make it easy. The first milk secreted by a lactating mother after the delivery, that the child takes in. It is advised that for the child to take mother's milk because of this colostrum. Okay, it contains the immunoglobulin. And which kind of immunity will it acquire? Is it active or passive? Yes, it is passive immunity, because this immunoglobulin is being inserted from mother's body to the child's. Okay. Next we are having immunoglobulin D. Next we are having immunoglobulin D, that is only 1 to 3%. The development of immune responses it is involved, and activation of B lymphocytes. Now the very important, okay, the very important, that is your immunoglobulin M, that is about 5 to 10% thick. They are the oldest, largest and the heaviest. That's very important. Oldest, largest and the heaviest and immunoglobulin is IgM. Okay, it is a pentameric structure, and so it is the largest one and heaviest one too. And it is the first antibody generated in response to antigens. Like suppose anytime, anywhere, any pathogen attacks to your body, the first antibody produced will be immunoglobulin M. Be clear about it. And one more thing, they show very strong agglutination. Okay, that's a very important point. A very strong agglutination. I told you about agglutination in the previous slide. Okay. Next, remember these things. Okay, just make a short note that you can recap. These are sure shot for AIMS. I'm telling you. I've given it. I got many questions from these. Okay. Next, immunoglobulin G. All right. IgG, that's very important. That is 75 to 80%. Most abundant, obviously, since they're 75 to 80%. Okay, the very important point which has a sure shot question is that they can pass placenta. Okay, now what placenta is, I think you all know. Placenta is something, what it is? Yes, placenta is a disc-like structure, okay, which helps in communication between the developing child, that is inside the embryo, and the mother and the carrying mother. Okay, so the only immunoglobulin that can pass this placental layer is the IgG, immunoglobulin G. It provides the immune power to embryo, and it is the lightest in nature. Okay, remember this because it contains a sure shot question. Okay, immunoglobulin G. Okay, now we'll be discussing further processes in the upcoming lectures. Till now, what we have studied is the very important part of the chapter, that is the basics of the chapter. Okay, you will be getting many questions from this. So keep revising, keep learning, and stay tuned to the lectures. We will be uploading many more lectures. If you have any doubts or if you are facing any problems, you can message me. You can contact me directly also. Okay, so keep learning, stay tuned to the lectures, enjoy this platform, Unacademy. Thank you so much.